The broadcast is now starting and all attendees are in listen-only mode. For your information, the session will be recorded and the recording may be made available to others. The participants list is in private mode so that other participants cannot see your name. The chat function is in private mode as well. Only the speakers and the moderator receive the chat messages. If you do not wish to continue based on the above, please log out. Ladies and gentlemen, hello and welcome to today's mindsON Centralized Statistical Monitoring Workshop, an initiative by Syntegrity's RBQM Academy. So today's session is the continuation of our last episode in September, how to use a CSM tool. I'm Tamara, the operations manager of the RBQM Academy at Syntegrity. This workshop will run for approximately 60 minutes. It is an interactive session that includes polls, brainstorms, and time for your questions and our answers at the end. So feel free to submit questions and comments for our speakers throughout the workshop using the chat box, please. Our speakers today are Johann Pröwe. Besides being a much sought-after industry speaker, Johann brings data-driven realism to the table, ensuring that clinical trial risk management goals are implementable in the real world. Johann has a considerable depth of experience in clinical research, which has been invaluable to many. This is Ting Yu from Syntegrity R&D. Ting Yu is one of our smart brains behind developing the myRBQM portal's risk recommendation system and pre-processing our clients' clinical study data. Ting Yu will engage you with an insightful demo of the myRBQM CSM tool. So, as already said, this is an interactive workshop. Later, I will unlock a toolbar for you that brings us to the brainstorming part that we have prepared for you. And in this toolbar, you can see an icon with a cup and pens. So when you click on that and next click on the T, it opens up a T box. And here you can pick a color and start writing. So for your information, your entries there are anonymous. Please don't be shy and brainstorm with us when it is time. As said, this toolbar will be unlocked later at the point where we are coming to the brainstorming section. Okay, thank you very much. So, and with that said, I would say we can start with our workshop here. Johann, the stage is yours, please.
Thank you very much, Tamara, for the introduction. So this webinar will start with a set of PowerPoint slides, and then we will go into a demo version where we created a set of different analyses. We, in this case, means Ting Yu, and those are the analysis that we would like then to share with you. A couple of slides you might think you have seen before, and we did that on purpose from our last CSM Part 1 webinar, because I thought, these are really, really important aspects, and therefore decided to reiterate those. So with respect to the centralized statistical monitoring, it is a way to look at data anomalies that were unexpected originally and not being considered in the list of key risk indicators. I've seen several cases where also, let's say, lab data or vital signs data had been used to create a key risk indicator, but those were really very, very specific key risk indicators for the indication or for the main efficacy criterion. But for the majority of the data, like the underlying hematology, clinical chemistry, and urinalysis, we usually do not create key risk indicators for those. So there are data that sit there, and those data can then nicely be managed and reviewed and analyzed via a centralized statistical monitoring tool. We will go into a set of different graphics that are available, and we will also discuss how the different data can be interpreted when you look at the different graphical displays. In some cases, I must admit that I like one or the other better, one or the other graphical display of the data. But that is really a personal preference, and therefore Tingyu will show you different ways of looking at the same data, and you then may have to decide what you personally prefer compared to your peers or to anybody else in your organization. What we frequently get asked is, now, isn't that something for a statistician? Isn't that a tool just for maybe a data manager or data analyst? But why should anybody else dare to look at these data? Because this is about statistics, this is about data distributions. And our response is usually, to be honest with you, this is something that anybody who has some natural science background will be able to interpret, will be able to draw conclusions from. And if not, then you still have, your statisticians, your statistical programmers, your data analysts available that may help you. But the majority of the data can easily be interpreted using the CSM tool. And as I said, we will demonstrate what such a CSM functionality can provide to the study data quality and the patient safety. So just that you get an idea of what is it that one can do with the centralized statistical monitoring. So I provided you with a workshop overview. Want to demonstrate what is the beauty of centralized statistical monitoring, what are the type of graphics that are available, what are the skills required, and then a demo done mainly by Tingyu. So the beauty of centralized statistical monitoring, you may have seen this chart over and over again, in particular the middle part of it. And the middle part is mainly around key risk indicators, maybe quality tolerance limits. On the right-hand side, though, you have different other aspects. But when we talk about centralized statistical monitoring, we are really talking about the analyses of pretty much numeric data that are available in your data sets. Those numeric data could be, as I already mentioned, lab data, hematology, clinical chemistry, urinalysis data could be used for vital signs data, ECG data, anything that is numeric and that could be used to demonstrate distributions, for instance, or relationships of data to each other. It could also be, just to name a couple of others, echocardiogram data, for instance, or in oncology, certain data sets could be used to use CSM. So, and this is a recap to what I already said last time in the first webinar. This centralized statistical monitoring has the beauty that it combines clinical monitoring with the exploration of statistical parameters of variables. So on the one hand side, we have the clinical monitoring aspect, but the monitors at the site in particular only see whatever the site generated as data. So if a site produced data for 5 patients, a site monitor may see data of these 5 patients. The CSM approach means that you can take a bird's eye view on the data and look at the data either for the entire study, for countries, or comparing countries against each other, or looking at the data from a site-to-site perspective. The beauty is also that you can do the analysis in real time. That means the moment that the data are available, you just kick off the CSM tool and you can see the issues that you might have in your data. You see the distribution. You may start mitigation actions, or at least trying to clarify these data. And again, just to make this 100% sure, we are not talking about edit checks here. We are not talking about, tiny little deviations that might have occurred. We are really looking at the bigger picture. The usage of statistical monitoring means that we review the data via complex statistical algorithms, and those have been recommended by Transcelerate to discover outliers or anomalies, and the results of which will inform the user about various monitoring recommendation and escalation options. So whenever you see such an outlier, you see data anomalies, you see bimodal distributions, then the question, of course, is what is it that I'm supposed to do with those findings? And we will address that in our last PowerPoint slide at the end of our webinar. And finally, the centralized statistical monitoring offers a single comprehensive overview for efficient cross-functional collaboration during the clinical trial process, which means that maybe a central monitor uses such a tool, but the central monitor then decides, well, this is a finding that I better delegate to a medical monitor, or this is something that I should forward to the data manager in charge of the data, or a statistical programmer, so that they can take a closer look at these data and initiate any corrective preventive actions if that is needed, or at least make sure that certain outliers are being reviewed again. So, because it's not necessarily the case that an outlier always means that there is a data error. It may well be that you have outliers due to the fact that the patient has a severe underlying condition. The patient is multimorbid, and therefore you find these outliers in your data. And Ting Yu will also demonstrate such a case, because we used a real-life data set for this webinar, and we found a couple of things that indicate that further investigation needs to be done. So, Tamara, I pass this on to you again. Is that correct?
Yeah, thanks very much, because this is our first brainstorming part here. The whiteboard is open now. Just click on the cup with the pens, please. Next, click on the T box. Here you can start writing. What do you say? What are your concerns related to the centralized statistical monitoring? What are your concerns related to the centralized statistical monitoring? Yeah, that looks good.
It looks good. It looks really good. Yes. Overkill. I like overkill. Always like overkill, because that's interpretation. Tamara, we need more
sort
space.
it a bit. Yes, indeed. So let's see how we can make it readable here. Yeah, I think that's good. Yeah, everything's readable. Okay.
Yeah, I think you need to stop it, because
All right.
otherwise I'm getting completely overwhelmed here. So what is it that, and everybody can see that now, is correct?
Yes.
Tamara? Yeah,
It's
okay.
available. Yes.
Yeah, okay, good. So on one hand, and I'll start here in the middle, the qualification of staff, the experience of staff, the knowledge of those reviewing, that is certainly an area that needs to be addressed. But on the other hand, I must admit that the data and looking at distributions of the data, and we'll show that in our live part of this webinar. the majority of the issues are really striking. You see that instantaneously without really having a detailed knowledge of what you're looking at. On the other hand, you're absolutely right in that an outlier should really be evaluated with respect to, is that potentially an issue, or is that just to be explained by the underlying conditions that this patient comes along with? So there's a certain balance that the qualification and experience needs to be taken into account. I also like this one here. The many findings can be confused with data cleaning, which is not the purpose of CSM. That is certainly correct. So data cleaning, at least in my opinion, data cleaning should happen prior to running, for instance, the CSM tool. But data cleaning, at least from what I experienced in my previous life, is usually focusing on individual data points, not necessarily at gross abnormalities or anomalies, sorry, not abnormalities, gross anomalies, strange data distribution, maybe also bimodal data distributions. I trained my data management organization in doing that, but that is not necessarily the case for the majority of the industry. So the majority in the industry is saying data managers focus on data cleaning of individual data fields, but hardly anybody looks across the data, across the sites, and takes this bird eye view. The ownership, yes, who ultimately is the one that should do it? We will get to this slide on the skills required. I personally think that I central monitor with some, let's say, good natural science background is certainly a good candidate, but also what has been called in the past the data analysts. So not necessarily those that do the data cleaning in data management, but people that have a little broader view of the data. They could also be good candidates and could assume the ownership for the central statistical monitoring tool. There must be someone from the quality management area because I see avoid unblinding here. And that is certainly a very important aspect. And I recall that our quality management people always were concerned about data managers taking a look at the data too closely and potentially unblinding themselves by slicing and dicing the data to an extent that eventually would unblind them. Certainly a concern. Depends on, of course, the type of data that you are looking at. And for instance, if the main efficacy data happen to be numeric data, one should have a process in place in an organization that those data will not be looked at using the centralized statistical monitoring tool. Because you're absolutely right, that could unblind the candidates. Then, of course, having the tools, the algorithms that one wants to apply. I have seen cases where really complex statistical algorithms had been used, and the interpretation then was pretty much left to the discretion of a statistician, not necessarily for a normal user. So the question is really, how complicated do you want to get, or do you want to have a functionality that can easily be interpreted and where you can easily decide, yes, this is an issue, or no, this is probably not an issue. And in the middle, I see that it will be leaned on too much, so critical thinking in the field will not be used. Yeah, but that's a common problem that we have in the industry, that the moment that we implement a tool, the moment that we implement an EDC system or an ECOA system, and edit checks in the system that people take a step back and say, okay, and the system is going to do it for me. That is absolutely not the case. So critical thinking is always required, irrespective of what people do. And I think, yes, I highlighted also this overkill. I would not implement CSM for a phase 1 study, for instance, because the likelihood that you detect anything that is not normal is very unlikely. And you certainly have other means in place where you would detect these anomalies much easier. So I would really use a CSM when you really have a lot of data available, a lot of data that you definitely cannot manage manually, but where you need some tools that help you to look at the data from above and then make your decisions. I think with that, I would like to leave it, but I'm amazed to see that you have a really good understanding of CSM already and the potential challenges associated with CSM. Appreciate your input here. Oh, Tamara, it's you again.
Yes, it is. So we have a poll for you. And I'm starting that on your screen now. The question is, when implementing RBQM, what are the most important features you are interested in seeing? If you could let us know, please, what you think when implementing RBQM, what are the most important features you are interested in seeing?
And I think you only can select one of the options, I hope.
Yes. I see a good majority for one specific answer here.
Well,
Interesting.
I must admit that I did not expect that. I, yeah.
Just another moment. Okay, we seem to be at an end. I'm going to present the results now. So here you are.
Okay, so everybody can see it now, correct?
Yes.
Okay, so amazingly, we only have two responses. Either you give me a RACT, so a risk assessment categorization tool in combination with KRIs and QTLs, and an alert system, though that means the basic risk-based quality management system. So 27% voted for that. And the rest wants it all. So give me a RACT in combination with the KRIs and QTLs, an alert system, plus the centralized statistical monitoring feature. I must admit that I did not expect that, but what that tells us is that there is obviously a need in the industry, at least those that participate in this webinar, to get a comprehensive system in place that allows you to tackle the potential risk from different angles and to look at the data from different perspectives. So that's really nice. I appreciate your input here. Okay. So just to briefly reiterate what type of graphics are available in the centralized statistical monitoring tool. And this is not a comprehensive list, so don't shoot the messenger here because we forgot one or two or three or even five other potential graphics. But these that we share with you here are the most commonly used, and in my personal opinion, they are the ones that probably cover 95 to 98% of what you probably need in your life. So usually what we see is, this pointer again, what we see is histograms, so something where you look at the data and the data distributions. My personal favorite are scatter plots, in particular when you look at two different types of data or two different data items. The interquartile ranges we have here on the right-hand side is also sometimes very helpful to see, where are the majority of the data and where are the outliers. Correlation diagrams, and we also have an example for that, can also be quite helpful, though I personally prefer the scatter plots over the correlation diagrams, but maybe they can be used in combination. So you start with a correlation diagram, and then you select certain correlations and run a scatter plot on them. Then Benford's law, which is usually used to look at certain data patterns or data manipulations, because humans are not that smart to really make sure that they use the various numbers equally. So that is something that we frequently see. And also heat maps could be quite nice to take a look at the data from the various sites and see, where are my outliers, where are sites that deviate from the others, or where are countries that deviate from others, or worst case, where are patients that deviate from others. So then the question is, what kind of analyses are you interested in? And there are different types of or levels of analyses. One can run such an analysis across the study just to get an overview, an overall impression of the distribution. And usually that is an analysis that is very helpful to just get an overview of what is happening in my study. Are there any aspects, any data that look awkward? And it also helps to get an impression of whether, for instance, if you happen to use 2 or 3 different central labs in different regions, do these central labs produce the same distribution of the data, or are they a little deviating from each other? Then you may have analyses between or amongst countries to identify country or regional differences in the data. And that is in particular important when you have, for instance, Eastern European countries and Western European countries, because you may see differences between those, let's say, regions, or even amongst those countries. And I've seen that in my previous life when looking at HbA1c data and diabetics, where the data distribution of the patients in Eastern Europe were really, really very different compared to the patients that had been recruited from Western European countries. You may also analyze data amongst the sites to detect site-specific patterns or anomalies, differences compared to all other sites. So that helps you to identify certain sites that are just different. And then, of course, going back to our brainstorming session, someone needs to take a look at, why is that the case? Is that the case because this site is different? Are they doing anything different? Or did they recruit, for instance, a completely different patient population, maybe still within the remit of the in and exclusion criteria, but they may be patients that are older, patients that are younger, patients that are sicker, that have more comorbidities, etc. So for that, it's also quite helpful to run an analysis amongst the sites. And then again, between and amongst patients at a site to detect very different distributions. So that can also be helpful. And once in a while, rarely though, we also look at patients or within patients to identify duplicate records. That depends on what our customers are interested in, but we even have an example. We found that by chance in the data set that we used. So another brainstorming, Tamara.
Exactly. The tool is open. What would you expect to be able to detect when applying CSM? What would you expect to be able to detect when applying centralized statistical monitoring? No expectations so far? Yes, okay. Very nice. I'm just moving the answers around here a little bit so they're readable. The trends over here, okay.
Everybody wants to detect fraud.
Oh, that's a good thing. We come to that in our next session, actually. Next webinar.
Yes. Okay, I think I have really a lot to discuss here. Let's talk about the outliers first. So that's the easy part. We have the outliers here on the left-hand side and trends that I think go into the same direction. Yes, absolutely. So outliers can be seen. But what I like is there are two types of outliers. Outliers in the sense of that the data are really very different from the pack of the patients. And there are outliers in the sense that I think someone wrote here, it's too consistent across patients and at a site. Too consistent means the data are too good to be true. And I like that in particular because we have this tendency in the industry that we always look at, the extreme values, and we have a tendency to forget about the data that are just too nice. They look all to be in sync with each other, and there's hardly any standard deviation. And that, in my opinion, is a good source to take a closer look at whether these data have been made up, so going into the fraud, misconduct area, or whether they happen to be that good. Yes, we definitely see carelessness and sloppiness, and we sometimes see data manipulation, in particular when we look at data that have been measured manually, not necessarily in the lab data, but in the vital signs we see that. Certainly data trends that are not covered by the key risk indicators could be triggers for clinical monitoring activities, in particular when you derive from centralized statistical monitoring that something looks weird at a particular site. And in such a case, you can then, of course, trigger a clinical monitoring activity. Anything that stands out from the rest is sort of the outliers. Detect issues early, and is that the purpose of risk-based quality management, to detect potential issues early and then be able to act upon it? Yes, and CSM falls into the same category. And definitely issues that cannot be seen at the site monitoring activities because the monitors just don't see it. So really appreciate that. A lot of good examples of what one would expect by implementing CSM. So just briefly on the skills required, and I mentioned that already. So at least someone should be able to understand the protocol of the respective study. So someone would have to read the protocol. That should be a requirement. Have a sound understanding of the expected data distribution. So for instance, what is it that you expect when you look at vital signs data? What is it that you expect when you look at lab data? And certain lab data like cholesterol, for instance, they are usually not normally distributed compared to, for instance, hemoglobin. And what is it that you can expect in the area of ECG data? And these data, for the most part, cover already, I would say, 70-80% of the data that one usually should look at. There are certainly other data. Sometimes you have echo data or you have any other data available, but the majority of the data are originating from these three areas. And of course, someone should have a good natural science background to understand lab units, for instance, and conversion factors. So when you look at certain distributions, oops, I understand what happened there. ECG interval relationships, and then linking lab data, vital signs, and ECG data to the underlying disease. So if someone has a hypercholesterolemia, then yes, you expect that patient to have a high cholesterol level, and not saying, okay, I have an outlier here, and therefore this is something that a monitor needs to check again. No, not necessarily. Take a look at the underlying conditions, and then you may get an explanation. And capable of distinguish between systematic and random observations. So if you really have a systematic different distribution in your data, then you should be able to detect that, or by looking at the distribution, understand whether this is systematic or whether this is something that just happened in one patient at a local lab. So I think these are the basic skills that I would expect someone that runs a CSM analysis. Of course, that individual should understand the purpose of RBQM, understand, if applicable, the underlying data structure. So if the setup of the CSM analysis is supposed to be done by that individual, you need to understand the underlying data structure. You need to be capable of transposing data set and knowing how to convert the data from one unit to another. So, when you have micromole per liter and you want to see the data in millimole per liter or in milligram per deciliter, you should have a conversion factor available. For this latter part, for this slide here, if you don't have the skills, then you should at least know someone who does and can help you to transpose the data or convert the data if that is necessary. And with that, I would like to hand over to Ting Yu and start the demo session.
Yeah, thank you, Johan. And let me share my screen. Sharing. Okay. Can everybody see my screen?
Yep.
Okay, yeah. So the first things that we want to go into is the hemoglobin example. So this is our demo environment, and we can create a histogram for the hemoglobin measurement from the lab data. And so let's start from the left-hand side. So this is the histogram that we directly use we directly use the data from the lab data. And as we can see, there's two distribution which widely separate from each other. And it is really hard for people to detect any outlier from this bar only. So this is something that people forgot to convert, or maybe the lab is using using different unit from each other, which might result in this kind of issue. And the even worse thing is that, for example, these statistics below, it's hard for people to tell whether this third or the first quantile makes sense or not. So so what we can do is to convert the data to the standardized unit. Therefore, we can be able to identify the minimum value, and certainly knows that this kind of value, like the minimum 4.6, could be an outlier. And other than that, we can see the bimodule distribution right here in the hemoglobin. So the bimodule distribution could coming from the histogram concentration or value is different between gender by gender. And then let's move on to another example, which is the within study comparison. So with this example, we are trying to talk about detecting the fraud or the sloppiness of the site. So for this part, we look into the trail digit distribution, try to figure out the fraud data. This method is commonly used in vital sign data, such as we demonstrate right here, which is the heart rate data. So the blue bar, as you can see if we're hovering around here, shows the last digit distribution of all the heart rate value in the study. And then the orange curve displayed the same distribution from one of the sites in the study. So this site we pick up is 1219. So as you might notice that there's a pattern in this orange curve already. So all the last two-digit value from this site are multiples of 4. So for example, right here is 68, and then the next one is 72, and then 76. So they are all multiples of 4. So the reason for that, we think maybe it could be the administrator probably didn't follow the protocol, which the measurement of the heart rate should take 1 minute. Instead, they measure it for 15 seconds and multiply the value directly by 4 to get the beats per minute. Therefore, if we're gonna to check the sloppiness, this is definitely a good choice for everyone to use applied on the heart rate side to detect this kind of fraud or sloppiness. Yeah, and then Johan, I think we can go together to the other example.
Yep. Maybe we start with the AST and ALT example where we used a couple of different types of analyses.
Yeah, sure. Let me click on this. Okay, we firstly go to the correlation matrix, which we expected these two things we pick up is a positive correlation. And yeah, so this is the first step we will do. So at least we can make sure that with our data, we are certainly sure that these two things are positive correlated. And then we will move to the scatter plot as Johan suggested in the previous slide, that after knowing they are positive correlated, it's nice that we try to detect the outlier from the scatter plot. So what we have here, for example, we can mark the data point with the label subject ID so that we will have a list of outliers right here. So for example, we might see the extreme values such as this data point, this red triangle, and it's a subject which is 240 with this scatter plot, and also knowing other extreme value as well. And all of them will be listed in this list, this table. Yeah. Johan, would you?
Yes, and what you can see is that the first two in the list are the same patients. So there seems to be a patient that has a fundamental problem with his or her liver, and therefore has really AST and ALT levels that are really, really high. And then we have a second patient there ending with the number 273, which is also in the hundreds with respect to the liver enzyme. So one could detect, those outliers, and then the expectation would be that someone takes a closer look at the underlying condition of that patient. Is there anything in the medical history or in the adverse events section that explains it?
Yeah, I think this also highlights what Johan presented previously about if we detect the outlier, sometimes it's not definitely an outlier. It's something that we should take a close look. Maybe it's a finding, but not something abnormally. Yeah.
Mm-hmm.
And that goes to the histogram of it. So the same as we show by the hemoglobin, we can still check these two with the histogram and to see the third and the fourth and the first quantile of it to see whether we can found the value shown is as we expected, so they are located exactly in the same range or in the normal range. And we can still find the abnormalities value right here in the histogram as well. So this is another method after we look into the scatter plot, knowing the correlation between two of them, and we can start with this histogram.
So there was a question, someone said, I would expect then in such a case where you have these extreme values that you see a medical history finding or an adverse event that corresponds to liver values. Yes, correct. That is also what I would expect. But it could be, that there is no entry for whatever reason, sloppiness, or the other option is that these values had been measured in a different unit, whether these are nanocartals or units per liter, these extreme values, or whether this is just a data entry error, worst case. If these data originate from a central lab, I would suspect that these data are okay, and then I would look for medical history or adverse events. If these data are from local labs, then I would dig a little deeper. Correct.
Okay. So moving on, let's switch to the second example. So the second example we have is the bilirubin and creatinine. So the process the same as we did for the previous example. We still go into the correlation matrix, and then later on we will look into the scatter plot, labeled by the subject ID, and the last one we can look into the box plot, maybe the box plot like this time. So for the bilirubin and creatinine, as we can see from this correlation matrix, it seems like not a really positive correlation. It's around like lower than 0.5, but close to it, I guess so. So and then it's good for us to investigate through the scatter plot to see what the distribution of that. Why is that not a really positive correlation as we expected? Once we look into the bilirubin and creatinine, and still we can use our knowledge to check whether those values make sense. And then other than this, we can still check the outlier, for example, these dot, which is around like 12.8 with bilirubin to see. And as we mentioned for the last one, still can check the medical history to see what corresponds to the medical history that was provided as well. Yeah.
And by the way, to you, this is a very same subject that we just looked at for AST and ALT, which also now has a very high bilirubin. So that is something that is really a problem for this patient.
Yeah.
And I think this was also the case where we found the duplicate records.
Yes.
I mean, there are other ways of looking at duplicate records, but we noticed exactly these two lines, that the values are exactly the same, which is quite unlikely, and maybe worthwhile taking a closer look at. And there's this other one, a couple of lines above, 13,
right here,
with exactly the same values. So one starts wondering, why is that the case? By the way, I just wanted to mention one should not have to just pick two analytes and then compare these two analytes against each other. The system should be capable of taking all the lab data and run the analysis against everything. Just for the sake of not complicating the analysis too much, we decided to select certain lab analytes and run the analysis against those two lab analytes.
Okay.
Yep, and maybe Tingyu, we take a look at the box plot case.
Yeah, sure. So the box plot here is labeled by the subject. So the box plots provide us an overview that what is the normal range of bilirubin or the majority range of the bilirubin and creatinine, which is helpful from the other angle, like compared to the histogram, we see the whole distribution. From the box plots, we will be able to identify some data point that is interesting for us to investigate. Okay, then let's go to the, shall we?
Yeah.
Yeah, yeah. Then we can move on to the filtration rate, or yeah, this one. Okay, so for the filtration rate, yeah. So the filtration rate itself is quite interesting to show here because we notice this two peaks in the distribution, which is something same as hemoglobin. So we notice a bimodule distribution, and this is something maybe can be explained by medic, or this is something interesting for us to investigate, like to decide or see whether this is caused by the gender, because this is a number that derived from a calculation. Yeah. Johan, do you have anything to add on this, or?
Yes, in particular for the glomerular filtration rate, we noticed that there are different underlying conditions. So, for instance, gender could be an important factor. There could be different races that uh, result in different GFRs. Or what also could be that there are different methods available. So, for instance, in the ECG area, we see that the QTC calculation, you have two different options to analyze the QTC, and therefore that could also generate slightly different distributions depending on which of the formulas you use. So one should always, and that's the reason why I said, earlier, the skills should be that someone has an understanding of natural science to be able to interpret these data, because otherwise we will probably generate lots of tickets or alerts or messages to the monitors or to a medical monitor saying, oh, we have a problem here in the data, while in fact it is just a matter of the distribution of the gender in the study. That's a good example here on the right-hand side.
I saw the chat saying maybe it could be the race. Yes, it could be one. It could be one of the possibility, right? Because as we can check the equation for calculating this could definitely be the same. Make sense, yeah. And yeah, and we are going to move on to the last example,
Yeah.
which is we use the ECG data and using the PQRST those interval to check the interval deviation compared to the heart rate. So how we calculate it is using, maybe we go to the histogram. So we use the heart rate per minute and using the equation to try to verify whether they measure the PR interval and QTC interval correctly. So the deviation right here is showing the millisecond. So the units right here is millisecond. So if you read there, so 125 is 0.2 second, for example.
Yeah.
And as we look into the data, we found, yeah, there's definitely a little bit deviations. So the more it close to zero, it's definitely better. But the deviation might come in from because we are using the heart rate to calculate the interval. So make the interval itself still got a time interval between one and another. So this deviation might coming from there, and from our perspective, 0.2 second as a deviation is quite minor. But what we'll suggest it is to look into the minors deviation, because there's definitely time between two interval, but it's not possible for the interval overlapping each other. So the minus one should have a further investigation. Yeah.
And by the way, Ting Yu, what also may have happened in this case is that if the heart rate has not been taken properly, then this type of analysis will also reveal that problem, that the investigators took the heart rate from their own measurements, while the ECG data came from a central lab or so. So there are ways to even derive data and then run the CSM on the derived data, because that is sometimes also quite helpful to detect any data anomalies. So
Yes.
I think that's another good example how to use CSM.
Yeah. Yeah, I think it circulates back to the example we showed at the beginning, which is the heart rate example. Not only using those within study distribution to check the stopness, this is as well a more advanced feature, but it's interesting to look into. Yeah. And this is the last example. So maybe tomorrow I will give the control back to you.
Yes, thank you very much. If you could stop sharing. Okay, thanks. And here we come back to our presentation. So the demo was that part.
Yep. Okay, and that is our last slide. So now we looked at all these CSM findings, and the question now is, what are the next steps? The next steps could be that you just accept what you see. And this is a slide, by the way, that we used also in the first webinar, but it's still applicable. So you accept what you see because there are good and sound explanations available. But what you could also do, and that is a functionality that such a CSM system should support, that you create a ticket for the respective function. And there, you get the link of the key risk indicators and the tickets or alerts generated by the key risk indicators. And the CSM system should be tied into that system that you use, such that you could also create tickets from the CSM environment, so that you have one consolidated set of tickets in your system. And the respective functions could be a site monitor to visit a site, as long as we talk about, for instance, site-generated data. Take a look at the central lab data and check their processes. So that should be someone else, maybe the study manager. The data manager should go ahead and convert lab data in particular to a common unit, whether this is SI or a common unit within your organization. And the medical monitor may have to be alerted about those cases that are outstanding. Consider adding a new risk to the risk library and to the risk log in case that is a systematic issue. Consider sending an auditor to a site. So if there is something that, in particular, the duplicate records that you might have detected, if that is a major volume of duplicate records, then send an auditor to the site. And what is really important is, though, document everything that you did. So that is probably the most important aspect, so that all the activities that you conducted have been documented. And with that, we are reaching the hour, and maybe there are still some questions that we will happily answer if that is the case.
Yes, we've already answered questions here, especially during the demo session. So at the moment, I can see no new question that has not been addressed. Feel free to put in your question in the chat window, please, while I'm talking here. I would like to